In our lab, we're interested in antibody-mediated rejection after kidney transplant. And so one of the problems with transplant is that you develop antibodies over time to the transplant and you can lose your transplant. So we're trying to find ways to eliminate the antibodies. Bortezomib is a proteasome inhibitor, and ixazomib is the next generation of proteasome inhibitor. This inhibitor is used in multiple myeloma to get rid of plasma cells, and it kind of essentially poisons them. And so the plasma cells go away, and the plasma cells are what are making the bulk of the antibodies. So we used a rat animal model in which we sensitized our rats and then did a transplant and gave them either ixazomib or didn't give them ixazomib. And what we found in our study, so this is a genomic study for this part of it. We looked at the up and down regulation of genes in the kidney. And what we were able to see is that some of the pathways that are involved in immunology, the T cell and B cell signaling pathway, and MHC class II molecules are all downregulated by the drug. So we're having the effect that we were looking for. We also looked at some cytokines called BAFF, and we looked at the actual amount of splenic plasma cells. Both of those are also downregulated in the treatment groups. We looked at serum IgG, which is an antibody. And we also looked at a deposition product, C4D, which is part of the complement cascade. When you have antibody-mediated rejection, it deposits in the kidney. So those are also downregulated when we give them the drug. So our conclusion is that this CNI minimization strategy, where we use some ixazomib and some cyclosporin, is able to mitigate the symptoms and mitigate the diagnostic symptoms of antibody-mediated rejection. This drug is easier to take than bortezomib. It's an oral, it's just a pill. And so we could possibly use this in antibody-mediated rejection in our patients. And there's currently discussion about clinical trials.